What's going on? My name is Sean Jackowitz, and the following is going to be a video of me presenting my research at the University of Aberdeen's Space Medicine Society conference. And it's an honor to be invited to give this conference. I similar talks recently at a Military Health Research Symposium where I was awarded by the Surgeon General of the Navy for the work that my lab did. It's been a blast to start speaking about this research. I really think it can be applied to a lot of really important areas of life, not just with space exploration and pushing the boundaries of human limits, but also with oncology and the cancer treatments that typically cause destruction in the body, as well as defeating cancer. There may be a method to protect against that, or some new methods to treat cancer, and then also with warfighters and our service members to protect them from the damages that they undergo or to enhance their natural abilities already. So yeah, if you enjoy this, it's been my life's work since I was 17 years old, 26 now. So if you have any questions, feel free to let me know. A biological spacesuit, ectopic expression of genome stabilizers for deep space survivability. That's what I'll be talking about today. This is my own personal research that I've been working on really for a long time. I have a lot to say, but I'm going to try and keep it down to the 10-minute timeline. Introduction. I'll be talking about risks to astronaut health in deep space, cell therapies, sides of current gene editing technologies, and mammalian artificial chromosomes. So what are some risks to astronauts in deep space? The Human Research Program at NASA has come up with a nice acronym for us, which is RIDGE, and it stands for Radiation, Isolation, Distance from Earth, Gravity Changes, and Environmental Limits. I will be talking specifically about radiation today. My research looked at mostly radiation, but in general, you can imagine any of these buckets can cause genome instability, damage to the genome in a variety of different ways, whether that's just fatigue or reactive oxygen species to damage the cell, but specifically radiation is a threat I want to cover today. So the radiation risk. The radiation risk of leaving the low Earth orbit is different than what we've experienced so far. It's definitely different than what we experience here on Earth, but even the ISS has been protected to a large degree from some of the more dangerous ions that you are prone to when you leave the magnetosphere. And specifically, there is a type of ion, an iron ion, that is both high charge and high energy. And the way that it impacts your DNA is almost like a grenade, and where shrapnel will actually fly off of your DNA, and these highly charged parts of your DNA structure will go on to do more damage. And so this type of radiation damage actually causes kinds of tumors, more carcinogenic tumors. And so there's an even greater need to try and prevent this kind of damage than just being in low Earth orbit and, of course, being on Earth. But the lessons we can learn, I think, can be applied in all three situations. And then when it comes to possible protections, there have been a lot that have been talked about already. There's a large literature of radiation protection. And to be fair, the solution for all of this biological spacesuit is really going to be a combination of a lot of different angles. Of course, physical shielding is a possibility, nutritional support, antioxidants, in the diet, pharmaceutical support. Gene editing is a bit more controversial. We're still not all together on where we want to be or whether or not it's ethical to permanently edit an astronaut's genome. And that's where I think my lab's technology can come in with cell therapies and editing a non-native part of a genome. And I'll get to that. So why don't we just use the technology we already have, right? CRISPR-Cas9 is fantastic. It does incredible edits. It's simple to use. But the problem with CRISPR-Cas9 is its off-targeting effects. We still haven't ironed that out. Base editors are, but all of these CRISPR systems actually select for p53 mutants. And you don't want to, if you're going to arm your astronaut with a cell therapy that's been edited via CRISPR and it's selected for p53 mutations, you don't want to then send them out into an environment that's carcinogenic in and of itself, even when you're healthy. That's not ideal. Viral and plasmid delivery options for like genetic payloads, that's great. They work very well, long history of using them, but the problem is that they are limited by their payload size and the complexity of the circuits you can actually build. And that's going to be we're going to need complex genetic circuits and genetic engineering to overcome a lot of these challenges that we're going to face in deep space. So these are two reasons why I think we're limited by the technology we currently have. So what if we had a platform that was capable of large and complex genetic payloads with genome stabilizing factors already pre-installed? That's where our mammalian artificial chromosome comes in. So our chromosome is about 60 megabase pairs. That's about the size of the Y chromosome. It offers complex and large range genetic regulation of gene expression, and it has a modular design. It's got about 70 integration sites. You can think of this a motherboard with 70 insertable like USB sites, and I'll show you a little bit more about that in a minute. But it allows you to build of very complex situations. The work I'll be showing today is only using one of these integration sites. It can get quite complex when you start thinking about genetic circuits that feed back on each other and expand over 70 integration sites. And this is all without having to edit the native genome. Our chromosomes do have kill switches, so if anything were to happen or if we wanted to uninstall them, that is a possibility. So none of the altering or engineering has to be permanent to the human. So could we make a mammalian artificial chromosome with inducible genetic circuits for ectopic expression of p53? That was my specific proof of concept project here. To go quickly through the methods, and I'm happy to expand more on this later, with the targeting vector design, I could talk like a whole conversation about just how to design these chromosomes, but I won't do that. I'm going to fly through them as I go through, but just to talk quickly what one of these designs look like. So already when we get one of our cells, in this case we're using CHO cells, or Chinese hamster cells, we transfect the chromosome inside of that using typical transfection protocols, and in the chromosome are these at P sites. These are the integration sites, and our vectors, which is a plasmid, have these at B sites. And when you transfect the vector with this enzyme, or ACE integrase enzyme, what you get is a recombination event, and your gene of interest you installed into the plasmid. That's how quickly it works. This is a very fast turnaround. Once you get the chromosome inside the cells, it's very easy to start integrating different plasmids with different genes of interest. And so my plasmid here has a TET inducible promoter. So that's a tetracycline promoter, turns on, if you will, in the presence of tetracyclines like doxycycline, which is what I used in my research. And then downstream is whatever you want. And here we're using p53. So our cell line is p53 deficient, and then we give it the p53 gene via our vector. So this is just verifying that I did indeed get the vector into the chromosome, which is in the cell. This is looking at the junctional PCR from the right side, and this is the left. The different mutants we're using are belonging to another research project, so I won't waste any time going into that, but just paying attention to the wild type is what's most important for this research. So this is our overall cell culturing workflow, very standard. Once we get our cell culture up to, our cells up to 70% confluent in a 10 centimeter dish, we separate it into 2 centimeter 6-well dishes, and then one group will be exposed to doxycycline and one will not. This is where we prove whether or not it was p53 we turned on. So we're looking at the RNA expression of the DNA. And so we use reverse transcriptase polymerase chain reaction. And this is just a quick schematic of what that looks like. So this is our control. I wanted to test whether or not we could actually still express proteins and genes. And so this is just a housekeeping gene. As you can see, both groups, regardless of doxycycline, can express their genes. And then this is what happens when we have one set of cells in doxycycline and another cell group not in doxycycline. As you can see, the wild type p53 is expressed when the cells come in contact with doxycycline in their media, and they are not expressed when they don't. Now, this slide actually shows some of our data from another project that we worked on that we showed at the American Society for Gene and Cell Therapies. And this was showing that not only can we do one circuit, but we can expand this to 2 circuits, and they can run both in tandem and oppositely, or not at all. We can turn it completely off, just depending on whatever inducible promoter we're using. So here we use tetracycline to induce GFP, And then we used Coumadate to induce RFP. And as you can see, we can do this both at the same time, at different times, or not at all. So, conclusion, MACs can be easily engineered with inducible systems to ectopically express genome stabilizing factors like p53 or any of the other ones you can imagine. This system is very quick turnaround. It took me about 6 to 8 weeks to finish this entire project. It's basic cell culture requirements, so it's not anything that couldn't be done on the IS itself. And then the MACs can be combined or can combine several inducible systems for complex genetic engineering while remaining stable, portable, and powerful. These artificial chromosomes can last through many generations and many cell divisions without any loss of fidelity. So it's something important when thinking about the long-term use of this and how durable this might be in someone as a therapy. So going into the future direction, all right, we can express p53 inside of cells at will. Does that actually add any benefit? So right now I'm testing whether or not the cells that do turn on p53 expression, do they actually then become more protected against radiation damage versus ones that don't? So that's where we are right now in experimentation, and I'd love to get that data and wrap it up into a paper. But then narrowing down on specific cell therapies, the data I showed you on the bi-inducible system, that was actually done in mesenchymal stem cells. So human mesenchymal stem cells. So we can actually do this in a stem cell line that leads to hemopoietic stem cells and all hemopoietic lines. And when thinking about cell therapy targets, I specifically focus on, or my thoughts are focused on the immune system and especially lymphocytes with how they're more sensitive to radiation damage, but also macrophages, megakaryocytes. We can get cell-specific targeting with these promoters. Only macrophages produce X circuit or only lymphocytes produce X circuit. Of course, I'd love to test this in spaceflight for modeling purposes and to see if it would actually work. Maybe there's something we haven't thought about that's unique about spaceflight. We do have mouse models, and bringing one of our specific cell lines into the mouse model would be a goal. And of course, eventually human modeling. Applications on Earth is for military members who are literally assaulted with radiation or reactive oxygen producing species. So I think this could be an equally protective measure for them. And then cancer patients, myelodysplastic syndrome is a symptom or is something that happens after getting treatment for cancer via radiation or chemo. And so could we protect patients from that and the variety of other damages that their body undergoes when receiving treatment? As far as what this would look like in application, we would acquire the cell therapy target from the patient. So that would be their mesenchymal stem cells, or we can honestly just use Yamanaka factors and create induced pluripotent stem cells from their skin. We've done this in the lab already. We then can characterize the strengths and weaknesses of their genome. As we gather more data about what are the threats facing the genome in space, we can take the astronauts' own genome and cross-analyze what do they have that might put them at more damage or at less damage. Can we leverage the strengths that they already have? We then take the cells we've taken from them, we give them the artificial chromosome, we then customize vectors to leverage those strengths and weaknesses, edit their chromosome, and then expand these clones so that they have their own bank. And this is the biological spacesuit, this own cellular bank that they can tap into and infuse back into the patient. And if we need to do several infusions, or if the infusion needs to be changed or re-edited, that can actually be done on the IS, because as I said, the process to do this is simple cell culturing. So yeah, that's the research. I want to thank the doctors that I worked with, Dr. Ed Perkins and Wei-Hyung Yang. Thank you so much for the opportunity to work on this project. I've been working with Dr. Perkins since I was 17 on the artificial chromosome project. So seeing it come so far and throughout my career has been really fun. And then we were funded by the NIH and my own medical school summer research program. And then these are my references. The end.
Thank you so much for that. That was amazing. If anyone has any questions, feel free to fire them into the chat now. We'll just give a minute for questions. I was going to ask though, you mentioned the artificial chromosomes have kill switches built into them. How do you trigger those kill switches? And is that something that like can only be done artificially, or could it ever be triggered by the environment?
So they wouldn't be done by the environment. There's a trigger for it. I can't give away that information, maybe for obvious reasons, just because that would be a possibility.
Yeah.
But the artificial chromosome itself was originally funded by DARPA, and that was one of their prerequisites, that if you were going to build this system in a human, it would have a turn off button, and it does.
And I was going to ask one other thing as well, if that's okay. Super fascinating presentation. Yeah. Sorry, let me collect my thoughts. And
You're good.
when you were speaking about looking at the genome of astronauts and then looking at the strengths and weaknesses and editing it, I'm just going to ask, do you think there's the potential there for over-diagnosis and like over-preparation and changing things that don't need to be changed? And do you think that's a worry or concern within that line of thought, or is that already addressed?
Yeah, no, that's a great question. And that is a concern. I think that's why our sort of the space medical community feels hesitant about a lot of the ideas behind gene editing itself of astronauts. Like, we don't know the downstream consequences of some of the gene edits that we would do to a person, maybe protect them against radiation in space, but like, what happens when they come back to Earth? Can you reverse that kind of thing? As you said, can you go overboard? These are all really good questions, and why we think this platform, using a artificial chromosome that's separate from your native genome, you could do a lot of the edits on that without touching the native genome. And like I said, with the kill switch, if you want to turn it off, or because of the way you make the circuits, they're only turned on at certain situations. When you're not using this chromosome, when it's not an environment like in a doxycycline meat, it's compressed, it's in a heterochromatic state, it's innate, your cell is not going to tap into it at all. Basically, what it consists of is mostly just satellite repetitive DNA, and then your circuit, whatever you inserted in with your gene of interest, and then more satellite DNA, and then another circuit, and it just goes on to that. So there's no other promoters tapping into it or anything like that. So we think that's a better way, this have your cake and eat it too situation, where you can edit the genome, but it's our synthetic version of the genome, so you don't have to go in and muck around with the human's actual genome and weigh all the pros and cons to that.